brain. Well, you know, that's actually somewhat harder to look at, but if it's in every cell in the body and it's in the SCN and it's in the olfactory bulbs, and it's actually, we now know it's in the hippocampus, I would put my money on it being in every cell, especially if it's in every cell outside of mammals. But interestingly, it needs to be orchestrated by the master clock in the brain. So when you lesion the SCN, you may remember behavioral rhythms went away. If you look at the liver or any of these other tissues, the rhythms go away. The measurable rhythms go away when you lesion the SCN. So what's going on? When you take the SCN out and put it in a brain slice and keep it in vitro, I showed you many examples of that, it just keeps on ticking. When you do this with any other part of the body, and this is part of what's going on in the U paper, the rhythms appear to disintegrate. They are no longer cohesive. But if you look at individual cells, they're still keeping time. So what happens is you are losing the coupling between the cellular components of the tissue. So the tissue clock, gone. You have no longer got coherence. Those individual cells are each ticking, many of them are ticking at a little bit different rate. And whatever it is that keeps them coherent in the body is gone once the SCN is gone. We don't know what that is. And furthermore, once the SCN is gone, you know the rest of the body no longer has information about when it's day and night or summer or winter in the outside world. So the SCN is the only portal to information coming in from the environment to the body. So this is kind of, yes, clock-to-clock coupling. I'm going to say a little bit about approach to clock-to-clock coupling that we've been working on, and it goes back to my telling you that the SCN is the most peptidergic part of the brain. This work was done with Jonathan Sweetler, and it's using a number of different of the innovative methods that he's on this campus. He's a very renowned analytical chemist, and what he likes to think about when he goes to sleep at night, I believe, is how could I measure something very small and local in the brain? The goal being to actually look at what's released from individual processes in the neuron. And that's not quite there yet, but it'll be there one of these days. But in this case, we used solid phase extraction beads, which are the kind of beads that you put in a column if you want to collect proteins and isolate them. And what his idea was is just to lay them on the brain. So actually, you know, one of my former students, Norman, did that. He laid these solid phase extraction beads. This is a fresh piece of rat brain. I showed you stained tissue from the rat brain. These are the optic chiasm. This is the third ventricle, and this is in the part of the hypothalamus in the region of the SCN. And these two little areas, they don't actually look that clear, but you can kind of see them here. You can actually see them in fresh tissue. There's nothing myelinated there. The fibers are very short. Basically, you're looking at cytoplasm and a little bit of membrane around them. And basically what he did there was to put these beads on, and some of them are in another nearby part of the hypothalamus called the supraoptic nucleus. This is what releases vasopressin into your body when you're hemorrhaging or when you're lactating mother, when you need extra fluids, or if you're eating lots of salt. And so this makes vasopressin. This is the optic tract. There is no vasopressin in the optic tract. And then this is the SCN, two subregions. This is the highly vasopressinergic part, and this is a different part. And then they stripped the peptides off of these little beads and analyzed them. And this is the pattern of vasopressin between 0 and 4 when the lights first come on in the morning, 4 hours later, 4 hours later, and so on. And you might ask, and actually using the HPLC methods that he uses, high purity liquid chromatography methods that he uses, you can separate every substituent in that sample out by its mass to charge ratio. And so what we have here, we know it's vasopressin based upon the mass to charge ratio, and you can also then break it up and sequence it, which was done here. So, you know, this is absolutely vasopressin based on all analytical chemical methods. And notice all these little peaks here. If we didn't have such a high resolution scan, we wouldn't see all those peaks. And so I was once put on the spot when he has a center from the National Institutes of Drug Abuse to study peptides in the SCN and other parts of the brain. The visitors asked me what those were, and I had to say I didn't know myself why they were different. It turns out they are the natural isotopes of carbon and hydrogen that we all live with, that are making the absolute molecular masses of these forms of vasopressin a little bit different, and you can distinguish them. That tells you how high resolution and sensitive this method is. And then in this case, we basically put the SCN in a chamber, collected the media, and analyzed it for peptides. And every 4 hours, just like we did here, we took the samples out and ran them through the analysis. And this is the peak. This is all of these data put together. This is the peak in vasopressin. It's a major released peptide. But then we saw several other peaks. Notice here. Interestingly, of the peptides that we picked up in this way, only 3 had previously been identified. And that was a big surprise. And so then we were faced with, well, what are those other ones? And we now know that one of them, little SACE, is a major peptide that no one had ever seen before. It's a major signal in the SCN for communicating light signals. But because they're released, the idea is that they can be coupling signals both within the SCN, between the individual clock cells in the SCN, because remember, even though we're measuring 24-hour oscillations in the whole SCN, it's still comprised of individual cellular clocks that are keeping time. But also signals that are going out beyond the SCN to other parts of the hypothalamus and possibly further. Now that's a very contentious issue, and that is how diffusible are peptides in the brain. And there's evidence that small molecules are quite diffusible in the brain, that depending upon how many membranes from other cells they run into, they can move really far from the site of origin. But it's not basically, most people don't think that's the case. You have to actually do the experiments to demonstrate it. So then the clock of the brain organizes behavior. Here's what I updated. It emerges from these molecular and redox feedback loops in cells. It integrates information from most brain and body systems. It's functionally dynamic like the cell cycle clock. And it gates input pathways restricting access of signals to the clockworks. I haven't told you about this yet. This is actually the next thing we're going to talk about. And that is how the clock basically shapes its ability to see signals coming in. I think I did mention it briefly, but not in very much detail. I would like to talk, could I talk, do you mind if I talk for a few minutes longer? Slim? No. Okay, because usually I talk till 5 and then we have a little break. So basically, I'm going to move into beginning to talk about this aspect of the gating of the signals that are coming in and the range of signals the SCN responds to. But I like to start out by asking you the question, why do we sleep at night? It's not a trivial question. Not everybody sleeps at night. Blind people don't sleep at night. And you might ask, why is that? So these are the learning objectives. What are the anatomical substrates by which light signals access the clock in the brain? How does light act on the clock to appropriately phase behaviors like our sleep-wake cycle? How does light signal to the molecular clockwork? This is the only target that we know about, actually. We don't know anything about light and redox. What is the significance of different responses to different Zeitgebers? Remember from our chapter in the book, Time Givers. And then how can alteration of clock elements cause sleep disorders? And this is a very interesting, fertile field. So basically, I'm going to tell you a little bit of a story, which is a good way to think about this. So this is a bunker. It's an underground cement cave, basically, in southern Germany that was built during the Second World War. The research institute that Jürgen Aschhoff, one of the 2 founders of the field that was talked about in the chapter that we read, his institute was nearby here, and he wanted to ask how people, humans, would respond to an aperiodic environment. Now remember, this is what we do with our experimental rodents. We put them in an environment where there are absolutely no time cues, as to the best of our ability to control it. It's hard to do with people. So what he did was ask for volunteers to go down and live in the bunker. Pat DeCourcy, who wrote that first chapter in the chronobiology book, did that when she was writing her PhD thesis. And I remember her saying, you know, basically, I took out my books, I went into the bunker. I believe they were required to leave their clocks outside. And of course, there's no reception for radios or phones inside the bunker. And basically, people were just asked to go ahead and, you know, live their life. They weren't asked to live in the bunker in complete darkness. They basically were asked to basically live according to their natural body rhythms. And this is what they found. And now what we're looking at is a human actogram. And so basically where it's yellow, this person is up, and where it's magenta, they're sleeping, and where there's an uptick, their body temperature is high, and where there's a downtick, it is low. And so they have two measurements, rest activity and body temperature. And notice, what does this look like that we saw before in one of the earlier lectures? Where did we look at behaviors in an aperiodic environment? It was in my first lecture. You remember the little rodent that we were actually sending to Hawaii? We had it entrained to a light-dark cycle, then we shifted it by 6 hours, and then we put it in complete darkness. It looked very much like this, where it started to run a little bit later. This person's getting up a little bit later each day. And it's going to, getting up a little later, going to bed a little later, and so is the body temperature rhythm. It's actually moving with the rest activity cycle. The period of this rhythm is a little over 25 hours. And these kind of data that first came from Ashoff were some of the evidence that humans basically have long rhythms, and most, as I said, most of us do. But what happens if you're basically running long by an hour each day? What happens after 2 weeks? Are you sleeping at night? Not if you're running an hour long. You're basically sleeping when it used to be day. And so this is basically living by our endogenous clock. This is the way, as I mentioned, blind people live. They don't have information about light in the environment, and they basically free run according to their endogenous clocks in the absence of information about this major signal in the outside world, light. So, yes, what is missing? What is missing is the external signal, the rising and setting of the sun, which does come up a little, I will admit, a little bit different time each day, but the periodicity of the appearance of light is, the sun has got a 24-hour cycle no matter how many hours of light and darkness there are. So what's missing is the daily alternation of light and dark, and this is just basically to tell me I'm going to talk about the substrates of this now. And this is a painting by Magritte, which I actually consider to be perfect for the concept that we're going to talk about here. And that is that the eye is not using information about shape or color or motion, the visual parts of what the eye does, to tell the clock what time of day it is. The clock has a completely different kind of access to light in the outside world. It is a specialized group of photoreceptors, and these photoreceptors are not, I don't know how many of you must have heard about rods and cones, basically the basis of black and white and color vision. These photoreceptors are actually the cells in the retina that project directly into the brain. They're the last place you get visual processing in the retina. Retina has many layers. These are the cells, they're basically on the toward the center of the retina, but they send fibers into the brain directly. And these cells, some of them, are directly photosensitive. They have a very evolutionary ancient photopigment called melanopsin. Melanopsin is in fish. It causes fish to change color, match the environment. Melanopsin is in this special class of the retinal ganglion cells. Did I perhaps say that on this slide? Yes, the retinal ganglion cells expressing melanopsin, and they are directly photosensitive. They have glutamate and a peptide called PACAP as their neurotransmitters, and they project to the SCN. And this shows you what these cells look like. The melanopsin is in the membrane. It's staining the cell body of these retinal ganglion cells, as well as the membranes of the fibers. And these would be the dendrites of the retinal ganglion cells that are basically spreading out in the retina and catching the light. So they're basically forming a network over this part of the brain. And then the axon, the information flowing part of these cells, is going into the optic nerve right up to the hypothalamus. And so actually, if you would have asked me 10 years ago, should I work on the retina? And I would have to tell you, there's a lot been done on the retina. I don't know how much really new there is to find there. There have been at least 3 Nobel Prizes awarded for how the retina works. And then these guys, how these retinal ganglion cells that are directly photosensitive have completely different targets in the brain. And interestingly, not the case in all areas of science. They're all friends and they all collaborate. Samer Hattar is at Johns Hopkins, and David Burson is at Brown, and Jens Hannibal, he's in Denmark. But they have done a very, very nice job of developing this story. I think only last month, Samar had a new paper in Science. One of my former students was on it. And basically, this will, I think, be my last slide. It's telling you it's so interesting about these retinal ganglion cells. Here are the cells I showed you. They have these very large, what they're called dendritic trees, where the dendrites reach out from the cell body. If you take out one eye, so that's called unilateral enucleation, but then you stain with, you've got a marker for, you're making beta-galactosidase, which you can stain for whenever the melanopsin is turned down. And so this gives you blue wherever melanopsins are expressed. The right eye was removed, but you see less staining on this side of the optic tract, but once you get to the SCN, it's about the same. So that means there's lots of bilateral projections there, because the one eye that's left is sending projections to both eyes. So it's sharing information. These cells innervate the SCN most strongly. This is right where they enter the brain, but they go on to multiple targets in the brain. So here's where this is what we're looking at here. Here's the eye, here's the SCN, and then there are other areas in the hypothalamus that they communicate with. They go on up to the thalamus and the parts of the brain that are involved in sort of extracting information out of what comes from the eye, and then even further up into other parts of the brain. So the reason Samar had a paper in Science about a month ago was he's still finding new things that these cells do. There are 5 different kinds of them now. And we've known for a few years that they mediate the pupillary light response. That's your response to bright light. They regulate the autonomic response to a flash of light in the dark. Your heart rate goes up. Your melatonin is suppressed. Remember, melatonin's made at night. And then the acute effects of light on movement and sleep. Remember what happens to a mouse when it's out in a field and you turn the lights on? Remember, they're running around at night. They freeze. They instantly stop moving. And you and I could do the same thing. And also on sleep, and what that new paper is, effects of light on mood. So, interesting. A whole new type of cell, whole new pathway. It's called the retinal hypothalamic pathway, or RHT, retinal hypothalamic pathway, bringing information about light into this part of the brain. And this simply tells you that if you knock out the rods and cones and the melanopsin cells, you basically, I guess, right, you knock out all those photoreceptors in the eye, and basically you don't get any entrainment of the SCN. And so I think I'm going to stop here, because now we're going to move on to talking about more of the light signaling pathway. And that's another story. So good. 5 after 5. We'll take about a 5-minute break, and then we're going to have a discussion for the next half.